The broadcast is now starting. All attendees are in listen-only mode.
Hi, everyone. We're just going to wait a couple of more minutes for people to sign up. We can see that there's a lot of people that are in the process of connecting. So we'll just wait about 5 minutes before we get started with our webinar today. Thank you for your patience.
Hi, everyone. Just for those who have just gotten online, we are still in the process of having a lot of new people joining on the call, so we'll wait a few, like a couple minutes before we get started. Thank you for your patience.
Welcome, everyone. Good afternoon to those of you who are in the Caribbean. Good evening to those of you who may be connected from Europe. And good morning to anyone watching from the Pacific regions. Thank you for joining us for this webinar on spawning of the grooved brain coral, Diploria labyrinthiformis, affectionately known as D.lab. My name is Anastazia Banaszak, and I'm a researcher at the National Autonomous University of Mexico and head of the Corallium Lab in Puerto Morelos. Joining me is Dr. Valérie Chamberland, research scientist at SICO International and Carmabi in Curacao. We are co-chairs of the CRC's Larval Propagation Working Group since its inception, and we are very pleased to have so many of you tuned in. Right now, I think there's about 211 people tuned in, so welcome. We have chosen this species because, especially for beginners, it is one of the easier ones to work with. During this webinar, we will go over guidelines for monitoring of spawning, collection of gametes, and rearing of larvae in ex situ conditions. Sorry, I'm having trouble changing my slide. Here we go. The goals of this session are to share with you the basics of the reproductive biology of this species, which is rather unique compared to other Caribbean species. Also, we have compiled all the spawning data that we could find because there are differences across locations within the Caribbean. Using this data set, we can provide guidance on when to monitor spawning for this species. We do not provide detailed methodology, as this has already been expertly covered in the 217 webinar available on the CRC website. Our presentation is in 3 parts. First, Valérie will cover the reproductive biology of D.lab based on her 7-year-long study in Curacao. I will then follow with the data set that we have compiled across the Caribbean with monitoring guidelines and predictions for this year. Valérie will then continue with the do's and don'ts of larval propagation for this species, and we will end with a Q&A session. For those of you new to GoToWebinar, we will give a quick overview. During the transmission, you are muted automatically by the organizers. You can type in any questions you have, and we will do our best to answer them during the question and answer session. But please be patient with us because as of yesterday, we had more than 430 people registered. So we may not be able to get to answer all of your questions. As we answer questions, we would appreciate it if you could delete them to make our question and answer more efficient. Be assured that any unanswered questions will be answered and posted on the CRC website. We are also very happy to receive emails from you to attend any further queries that you may have. I will now turn over to Val for the next section of the talk.
Thanks, Anya. If you can make me presenter, I will now share my screen.
Yes, I will do that. Just need to get my mouse to pay attention to me. There we go. Okay.
Okay, Anya, can you confirm that you are now seeing my screen?
Yes, I can.
Perfect, thank you.
There you go, perfect.
Oh, hi everyone. As Anya mentioned, I'm super excited to be hosting this webinar today, and even more so to have captured so much interest from you guys. This is really great. So in this first section of the webinar, I will be presenting on the general characteristics of the reproductive biology of D.lab, I will be using a data set that we've collected over 7 years of observations on the southern Caribbean island of Curacao. And the results I will be presenting here are really a collaboration between the Comabi Marine Research Station on Curacao, SECOR International, Juan Sanchez's lab Biomar in Colombia, as well as the Pittsburgh Zoo where Skylar Snowden worked at the time, and the University of Amsterdam as well. But building this data set was only made possible with the help of many, many students, interns, and volunteers who helped with all of this monitoring data. So back in 2012, we were looking into expanding the range of species that we could use for restoration work via larval propagation. At the time, Caribbean coral spawning work really revolved almost exclusively around endangered species, such as the acroporids and the Orbicella species, for example. At the time, we were really hoping to add potentially sturdier species to the few corals that we were already working with. And well, ultimately, we really wanted to increase the success of larval propagation efforts, and especially on more degraded reef areas where more endangered or sensitive species might not be appropriate for restoration. So we consider brain corals as potential candidate species because, well, on many reefs across the Caribbean, they are still quite abundant, and that is also even on degraded reef areas. Our motivation to work with brain corals was even further increased by a study that was carried out by Mark Vermeij and Rolf Back on Curacao in 2005, in which they compared the abundance of juvenile corals to that of numbers that were recorded in 1974. They reported an overall very dramatic decline of about 50% of the absolute number of juveniles in those 30 years. Which, yes, was really dramatic. But on the other hand, there were surprisingly some species, such as, for example, Sidrastrias and Montastria cavernosa, which were recruiting in higher abundance on modern reefs compared to 3 decades ago. Then that positive trend was also the case for Diploria species, which, well, at the time still included Pseudodiploria clivosa and strigosa as well. So these results from Vermeij et al. Actually suggested that Diploria species might be like really ideal candidates for larval propagation work because they're actually still successfully recruiting in the current environmental conditions that might be harsher than before. So within those different brain coral species, we chose to start working with Diploria labyrinthiformis in 2012. What was known at the time about the reproductive biology of the species had been documented mostly by Ernesto Will and Wilma Vargas in Puerto Rico, as well as Elvira Alvarado's team in Colombia, and for the most part consisted of information that was issued from histology studies. So in their histology slides at both locations, it revealed that the species is a broadcast spawning species, and it also produces both male and female gametes, so it's a hermaphrodite species. But the most surprising finding was that they found that during gametogenesis, gametes would reach maturity somewhere in April and May, and that was followed by the absence of gametes in mesenteries somewhere in May and June, which all indicated that spawning of this species would occur in the spring. This is quite contrasting to many, well, to most, well, all other Caribbean broadcast spawning species that we know that are known to spawn either in the summer or in the fall in the Caribbean. In Puerto Rico, this was like spawning in the spring was confirmed by Ernesto's team, who placed overnight collection devices over colonies and recorded gamete release in the spring during multiple consecutive years. It was, however, in May and June 2011 that spawning of the species was first witnessed in situ in Bonaire by Ellen Mueller. Ellen Mueller, who, by the way, is an amazing underwater photographer. She's amazing at finding any spawning in different creatures, whether they're fish, inverts, crabs. I highly recommend visiting her website if you're interested in seeing more spawning of other creatures. But that said, so her observations revealed another quite intriguing characteristic of Diploria spawning, which is that it's a daytime spawner. So it basically releases gametes an hour preceding sunset, which again is quite contrasting to other Caribbean species, which are all known to spawn during the night, so after sunset. So building on these 3 different publications, we, on Curacao, so we were able to narrow down predicted times of spawning to the hour that was preceding sunset, somewhere between 9 and 14 days after the full moon in the spring. So in 2013, we put in place a long-term monitoring program of a population of 40 different colonies at Holiday Beach. Those colonies were randomly selected, and we identified them with permanent number tags so that we could track them over time. And then starting in April, we monitor every month the spawning activity of those 40 colonies. So while we thought we would be monitoring spawning for probably 3-4 months max to cover the spawning season of the species, Diploria basically spawned every month starting in May all the way till September. So we actually found ourselves monitoring monthly from April all the way to the end of October. That was really remarkable. So the general patterns of spawning of the species on Curacao are that, so there's basically 2 peaks of spawning. A big one in the spring in between May and June, and another smaller peak in the fall in August. And that resulted in 6 different reproductive events on that year. Spawning, similar to Bonaire, was recorded between, like in the hour before sunset, but there was a peak between 15 and 45 minutes before sunset time. So what does it look like when the species spawns? Here's a video that was recorded by Benjamin Mueller in 2012, which, well, I think describes pretty well spawning of this species. The first thing you might notice is that butterfly fish pretty much give it away. So on the days of spawning, they will really start swarming around colonies that are about to spawn, and they will start feeding on the gametes that are being released. Another thing you might have noticed is that the moment that those sperm egg bundles are visible at the mouth of the polyps, they will be released shortly. So compared to other species, the staging or setting time of those bundles is really short. So basically, once you see the gametes staging, they will be released very shortly. The other interesting thing about Diploria labyrinthiformis is that colonies might, they don't only spawn in one big burst, like we've pretty much seen in this video, but some colonies will start spawning in multiple pulses. And each of those pulses will last a couple minutes up to 10 minutes, and can sometimes be separated by intervals as long as 20 minutes. So once you start seeing a colony spawning, you might want to hang around because it might be spawning more in a few minutes. So following the 2013 monitoring efforts, we did continue monitoring those same 40 colonies until 2019. Not every month, but here and there periodically during the years, which resulted in a data set that spanned about 7 years of observations. And this data set actually allowed us to reveal something quite remarkable about this species. So in fact, colonies that spawn in the spring would never spawn in autumn, and vice versa. And that's with the exception of those 2 colonies in orange, which spawn in the 2 different seasons out of 29 colonies. But for the most part, those colonies are very consistent in their reproductive time. And on Curacao, this is so consistent that we're actually now able to predict exactly which colonies will be spawning which month. And it's almost making spawning collection efforts almost too easy, but yeah, very convenient. So besides this convenient aspect of those 2 reproductive cohorts, this segregation between the spring and autumn spawners started making us wonder about the possibility of those 2 different cohorts being actually genetically isolated, because the chances of gametes or genetic mixing between the 2 groups are very small. So at that time, we decided to partner up with Juan Sanchez's lab, Biomar in Colombia, to run genetic analysis. The results that you see here were generated by Matias Gomez Corrales, who I think is actually on the call with us today. If you do have really like more detailed questions about specific genetic analysis, I will be happy to direct you to him after this presentation. But basically the point of this slide is to show that these analyses really revealed that the two reproductive cohorts are indeed genetically isolated populations. And they're actually so distinct that they may actually form two different cryptic species. And that's despite that they look quite identical. They occur in sympatry, so they're on the same reef, same depth, and they're all intermixed together. So following this study, we have been compiling data from across the Caribbean, and with the goal to determine if there are similar patterns of spawning in this species in other locations. We also were curious to know if these two cryptic species are widespread across the Caribbean basin. So far, it seems like those two different cohorts are not present at all locations, but there definitely is still a big lack of monitoring data to be able to confirm this. So all of these different observations will be will be covered really in detail by Anya in the following presentation. She will go over all of these observations at each of those localities, and she will also be providing guidance to monitor spawning of this species. Before I pass it on to Anya, I would just like to add that any additional observations, whether they are positive or negative, would really help us detangle this puzzle. For us, this is quite important because the presence of such undetected species definitely is a roadblock to preserving biodiversity. So in other words, we cannot protect species we don't even know exist. So on these few last words, I will turn it on to Anya again for the next presentation. Thank you. Anya, have you received the invitation to share your screen?
Yes, I just did. Thank you very much.
Fantastic. Welcome.
Thanks for presenting those really interesting data. And obviously, there's so much work behind all of your efforts that have gone into understanding this species more. As Val mentioned in this section, I will present the current data set that we have and the monitoring guidelines, as well as predictions for D.lab spawning for 2020. And before I go on, I would like to thank the members of my dream team who are listed there, and many volunteers and students who aren't listed, who have helped with monitoring spawning collections and culturing D.lab in Puerto Morelos. And that's been with the help of SECOR International, the Mexican funding agency CONACYT, and the Puerto Morelos Reef National Park staff. So what is being presented here today is also due to the incredible efforts of teams around the Caribbean, particularly from Curacao under the lead of Valerie, and in Bonaire with Francesca Virdes of Reef Renewal Bonaire. We also have data from Rita Seyares and Maria Villalpando from Fundamar in the Dominican Republic, from Dr. Valeria Pizarro of the Nature Conservancy in the Bahamas, from Kimani Kitson-Walters from the Caribbean Netherlands Science Institute, and Eric Hutepen at the St. Eustatius National Park, as well as Dr. Elvira Alvarado from Jorge Tadeo Lozano University and Icomares in Colombia, as well as Skylar Snowden from Sea Life Arizona in the USA. I'm sincerely grateful for all of your efforts to date and your willingness to share data with us, to share with the public. So first, I'll go over the general patterns of Diploria spawning across the Caribbean region, of Diploria labyrinthiformis spawning across the Caribbean region. D.lab is pretty unique, and most of the information here I have summarized from Anna Jordan's master's thesis. Anna's supervisor was Nicole Fogarty of the Reproduction and Evolutionary Ecology Fogarty Lab, who at that time was at Nova Southeastern University, but Nikki is now at the University of North Carolina at Wilmington. So because D.lab is unique, we figured that we would spend a little bit of time talking about this. In this figure, you can see that it has a broad spawning window from May to September, with peaks in June and in August. However, complementary data from Curacao shows that there are actually two peaks, one in the spring and a smaller one in the fall. And the months vary depending on when the full moon falls within the year. So they can spawn basically potentially from late April to early November, depending on when the full moon falls. If the April full moon falls after mid-April, you may detect spawning in late April. However, if the October full moon falls within or after the third week in October, you could potentially get spawning in late October or early November. So there are peaks in May, June, and August, September. One of the conclusions from Anna's thesis was that that D.lab is the only species to spawn exclusively before sunset, and that it has the smallest range of spawning times, from about 117 minutes before sunset to 0 minutes before sunset. As you see here, I've used a negative sign to indicate the number of minutes relative to sunset, or RS. So the negative 117 means 117 minutes before sunset. And the peak was shown to be from -52 to -40 minutes. However, on Curacao, the spawning observations have shown that spawning can last until 10 minutes after the sun sets, with a peak ranging from 15 to 45 minutes before sunset. Some additional information that we get from Anna's thesis is that spawning dates range from 8 to 13 days after full moon, or DAFM, and with a peak between 11 and 12 days after full moon. But this can vary depending on location. For example, in Puerto Morelos, the peaks are on the 10th and 11th day after full moon. In her thesis, Anna has mentioned that there was no split spawning recorded for this species, which means that there's no spawning occurring in consecutive months in the same region. However, in Curacao and in Mexico, we've seen that these colonies that have been tagged, can spawn on 3 consecutive spawning months in Curacao and 2 consecutive spawning months in Mexico. So in the next slides, our attention will turn to current monitoring data. We'll basically go approximately clockwise around the Caribbean, starting in Florida. The only data that we have access to at the moment was collected by Skylar Snowden, Corey Walter, and staff from the Moat Marine Lab in 2015 in May, 10 to 14 days after full moon. The red color indicates that no spawning was observed on those dates. So based on this one set of observations, there may not be a spring spawn in Florida, but really we need more data to know if these are spring and or autumn populations in the Keys. From Eleuthera, we have data from Valérie Pizarro at Cape Eleuthera Institute, which shows that in 2019, colonies spawned in May and June, as indicated by the green color. So we likely have at least a spring spawn in the Bahamas, and it would be great to get more data to see if we have a fall or autumn spawn as well. In St. Eustatius, Kimani Kitson-Walters from the Caribbean Netherlands Science Institute and Eric Hootepen from the St. Eustatius National Park did not see spawning in the months of July, August, and September in 2018. So this suggests that there's no autumn spawning, but again, it would be great to have more data to verify this and to determine if there might be a spring spawning group there. In the Dominican Republic, Rita Segares and Maria Villapando of Fundamar have supplied us with a 3-year data set where we see that there are spring and autumn spawns at approximately 10 and 11 days after full moon. It is important to point out that the May and June spawns are the biggest spawns, and later in the year they tend to be very small spawns or even just dribbles. So we can see that there's quite a bit of variation from year to year and month to month, but there does seem to be a spring and autumn spawning groups. We also have a 6-year data set that was compiled by Ernesto Weil and Wilma Vargas from the University of Puerto Rico in La Parguera. And this shows that there also appears to be a spring spawn in Puerto Rico, and histological studies suggest that there is no autumn spawn. Data from Colombia in the Parque Nacional Corales del Rosario, collected by Dr. Elvira Alvarado from the Jorge Tadeo Lozano University and Icomares, suggests there is a spring spawn, but we would really need more data to know if there is an autumn spawn. Most recently, she found spawning in June, 11 and 12 days after full moon in 2019. During Val's presentation, she has shown that using long-term data sets from Curaçao and Bonaire, there are spring and autumn groups spawning in both of these sites. And recently, Francesca Virdis detected spawning in D.lab in April this year, which is really fantastic news. In Puerto Morelos, Mexico, we appear to have an autumn spawning group. Although we have gone out during spring, we have not detected spawning so far. Spawning appears to be restricted to July and August, and possibly September at 10 and 11 days after full moon and about 45 to 50 minutes before sunset. So to summarize all of this information, especially those of you who do not yet have data for your particular location, we suggest planning to go out from April to November. In April, if the full moon occurs after mid-April, and in November, if the full moon occurs after the 3rd week in October. We suggest going out 9 to 14 days after full moon and planning a 2-hour dive, where you're in the water about 115 minutes prior to sunset and staying till about 5 minutes after sunset. And just remember that even if there have been some negative observations in your region, spawning still might happen. So from the current data set and the information that that Val presented, it is obvious that we really need more monitoring efforts for this species. If you are in a position to be out in the water, we have included this table of predictions for D.lab spawning from May to November. You probably saw the beautiful full moon last night, so our predictions indicate that you should be in the water from the 16th to the 20th of this month to cover any possible spawning window. Please note that in July and in September, the full moon is reported as the 5th and the 2nd, respectively. However, the maximum brightness is reported in the minutes or couple of hours after midnight. So really, these are the nights of the 4th of July and the 1st of September. Therefore, We are counting our days from full moon using the dates July 4th and September 1. Also note that October has 2 full moons. The second one is on Halloween, so there could potentially be November spawning. So in the last section of my part of the presentation, I would like to give you indications for more material that may be useful to you. On the CRC Larval Propagation webpage, we have a downloadable PDF on the spawning monitoring guidelines, which have been adapted from the webinar that Kristen, Valerie, and Nicole gave in 2017 that is available on, to listen to on the CRC website. Also, you can download templates for spawning data in Excel. We have templates for slates that you can take into the field, and also for registering your data in a spreadsheet and developing a database for your area. We will send out a poll at the end of this webinar and would appreciate it if those who are planning on monitoring D.lab spawning could fill it out. And if you do have spawning or not, send those data sheets to Valerie at that email address. Because for us, it's just as important to have positive spawning data as well as negative spawning data, because that helps us a lot to try and be better at predicting spawning windows. Also, Nicole Fogarty formed the Coral Spawning Research Facebook group, which has now over 2,500 members. So congratulations, Nikki. This has been a fantastic effort and a really great place to share information on spawning and to upload spawning monitoring data. So those of us in the field can pretty much immediately know where spawning is or is not happening. So again, the email address for emailing spreadsheets is with Valerie. And finally, we are developing a coral spawning database. So all of the spreadsheets that you share with us will end up in this database. And it's through my university, which has a marine informatics unit called UNINMAR. The database will be public access. It's triple backed up to ensure that we don't lose data. It has a dedicated database manager for this particular unit who's experienced in running marine databases. The site is bilingual and it has the same basic appearance as the Excel spreadsheets that you can download from the CRC website. We are using drop-down menus to try to reduce error, especially in spelling and GPS coordinates. And our plan is to enter all of the spawning data that we currently have and open it up to the public. There will be different levels of access to ensure database safety. And if you have any questions, please feel free to email me. My email address is at the top of the slide. So thank you very much for your attention, and I will now pass the microphone over to Valerie.
Thanks, Tanya, for this incredible overview of all the spawning data from around the Caribbean and for sharing those guidelines. We're just in a process of changing presenter. Hold on. There it is. Anya, can you confirm that you are now seeing my screen?
Yes, I can.
Fantastic, thank you. So in this presentation, final presentation, I will be providing guidelines to the larval propagation of D.lab. And most specifically, I will overview the do's and don'ts to optimize your larval rearing process for this species. So first question that might come up to mind is why working with D.lab? As you've seen from previous presentations, the species may spawn up to 7 consecutive months in the year, which really significantly increases opportunities to do coral spawning research and restoration work across the year. And that's especially true in the spring, during which we are not caught up with mass spawning of other species like in the summer and the fall. It's also a relatively easy species for gamete collection because it's quite common on many Caribbean reefs. It's also quite easy to identify, and well, it's also daytime spawning, so it avoids the night dives. In terms of larval rearing, compared to other species, it's also easier to successfully rear the species because the embryogenesis of D.lab is very short. So the amount of time that they need really intensive care is quite shorter than for other species like the acroporids, for example. The larvae reach rapid competency to settle and usually do so at quite high settlement rates. So normally this species allows to generate quite a lot of successful settlement. So we highly encourage you to start with the species, especially when you are less experienced with larval propagation work. So during this presentation, we will go over step by step for tips to improve gamete collection, fertilization, and embryogenesis. We'll give insights on optimizing culture maintenance, as well as settlement and post-settlement survivorship and health. But please keep in mind that these are going to be really only pertaining to the specific species, and that guidelines given here might not be applicable to all species throughout the Caribbean and other regions. If you're actually looking for very detailed instructions on corresponding techniques, I once again would like to refer you to the 2017 webinar on Caribbean corresponding research and restoration, which is available on the CRC's website as well as the TNC's Reef Resilience Network website. So during gamete collection, a few tips. Stay alert to butterfly fish activity, because in general, that will really help you pinpoint which colonies are about to spawn. However, I would like to also say that you shouldn't rely on it. It could be that, for example, on your reef, there isn't a really abundant population of butterfly fish, and you might just not see that behavior. So yeah, stay alert, but don't rely on butterfly fish to tell you which colonies to collect from. Also remember that colonies, individual colonies on one night might be spawning in multiple pulses. So if you're monitoring one colony and collecting from that one colony, it's a good practice to keep an eye for additional spawning pulses before you would remove your collection nets. If it is possible at your location, try to track the same population over time. That way you might be able to start to detangle spring versus autumn spawners, which will really help you being more efficient at finding your colonies every spawn collection spree. We also highly recommend using a spawn collection net only once or on only one colony to limit the spread of the stony coral tissue loss disease, but also other pathogens among colonies. So it's not recommended to use the same net on multiple colonies. You'll be diving during daytime, but really close to sunset. So remember that darkness might actually sneak up on you. So please bring a dive light for your own safety. In terms of general characteristics of the gametes of D.lab, well, they release pretty large bundles, and those are about twice the size of Acropora palmata. They contain many, many tiny eggs, which are about 10 times more eggs than within an Acropora bundle. And those eggs are really, really small, so about half the size of Acropora eggs. So just to give you kind of a ballpark idea of what that means, is that in that one collection tube down here in the first photo, which is about 15 to 20 mil of spawn collection, you probably have something around 200,000 eggs. So that just means you shouldn't underestimate your collection, even though it might feel like a small collection at the beginning. D.lab bundles actually break up quite early after spawning, so usually within the hour. So that will typically happen during your dive. So don't worry that it's happening. It's actually quite normal. Another sort of fun characteristics about D.lab is that the pigmentation of eggs is really variable. So it can range from pigmentation of dark brown to purples, pinks, and yellows, actually super pretty. So just don't mistake that for a bad collection. It's normal in this species. Okay, so once you've collected your sperm and egg bundles from colonies on the reef, you will be returning to the lab, and it will be time to start initiating fertilization. So last year on Curacao, we conducted different assays to see if we could optimize fertilization protocols in a different range of coral species. Our general questions were, so how long after spawning do we still have to mix gametes together and still obtain a viable or a successful fertilization? We also were wondering what was the optimal sperm concentration for fertilization. And our last question was about the duration of fertilization. So we didn't know if there was an optimal amount of time that male and female gametes should remain in contact to allow fertilization. So all of this work was conducted by Tanya Dublado-Speck and Matthew James Bennett. Just again, I wanted to say a huge thank you to both of them for their amazing work. So in the next slides, I will be presenting the results of those 3 different experiments that they did. So the first experiment consisted of mixing gametes at different time points after spawning. The results show that there is a breakdown in fertilization success when gametes are somewhere between 3 and 5 hours old. This means that at that point, either the eggs or sperm or both are no longer viable, and that results in fertilization failure. So practically, for those who will be collecting gametes, this means that there is sufficient time after spawn collection to mix gametes together. Obviously, the better you mix, the earlier you mix them, the better. But you shouldn't be rushing and stressing about mixing those gametes. I think here the key is really to be slow and steady. Take a good breather. Once you're back in the lab, make sure you get focused and zen again before you start all those manipulations. In their second experiment, Tanya and Matthew look into the optimal sperm concentration for fertilization. What they found is that there is a breakdown of fertilization at around a sperm concentration, well, somewhere between 10 to the 6th and 10 to the 4th sperm per mil. So practically, this means that it's not recommended to dilute the culture below 10 to the 6th. I know that this number might sound a little hard to grasp if you're not really used to working with coral spawning, but typically a culture that's around 10 to the 6 will have a coloration, a bit like on the picture here. It will have a weak lemonade coloration. This is how we usually describe it. So you don't want to go much lower than that weak lemonade coloration for your culture. We, however, also don't recommend keeping concentrations that are really high. In their study, Tanya and Matthew didn't do concentrations higher than 10 to the 9th, but we can already see that fertilization rates were starting to drop a tiny bit after 10 to the 8th, and that might be due to what we called polyspermy. So that's the case when one egg gets fertilized by more than one sperm at a time, and then they're therefore no longer viable. So basically, if your water column is super murky, you want to dilute it, but don't go too dilute past the weak lemonade coloration. In their third experiment, they investigated the amount of contact time that was necessary between sperm and eggs to allow fertilization. Surprisingly, what they found is that fertilization success remained really high, even if the gametes were in contact for a really short amount of times, so as short as 15 minutes. These results have 2 implications. Well, first, a really convenient one is that on the night of spawning, manipulations in the lab go much faster because typically we would leave those sperm and eggs to fertilize for at least an hour and a half, or at least when we would see the first cell divisions. So by leaving those gametes together for a shorter amount of times, we're increasing the speed of those manipulations in the lab. The other implication is that by stopping fertilization earlier, we will be able to rinse and handle those eggs before they are undergoing the first cell divisions. And that's important because those eggs are, when they are ongoing cell divisions, are really fragile, and that's when we want to avoid manipulations. So generally speaking, as a take-home message for this slide, to be conservative, we do recommend allowing fertilization for at least 30 to 45 minutes. This is really to take into account that fertilization might take longer to take place if you, for example, are working with lower sperm concentrations. In this case, this experiment was completed 10 to the 6. But if you're working with lower sperm concentrations, it might actually take more time for sperm to find the eggs, so you don't want to rinse them too early either. This could also be the case if you have compatibility issues between different pairs of parents in your culture, so that one parent takes more time to meet the other parent as they are searching through for successful mates. So yes, again, we recommend here starting to rinse your eggs around 30 to 45 minutes after fertilization. So this slide illustrates a timeline of embryogenesis and is accompanied with different recommendations for culture maintenance and handling. And again, this is really pertaining to Diploria labyrinthiformis. Other species will have different timelines. For example, Diploria labyrinthiformis will have completed development within 12 to 13 hours, which would normally take for Acropora pomata up to 3 to 4 days. So again, please make sure that you only apply these recommendations to Diploria labyrinthiformis specifically. So here, if we first look at photo A, is within about 30 to 45 minutes after fertilization, we recommend rinsing the eggs, and that's to discard all of the sperm that was not been used to fertilize eggs. We typically do this using fat separators, or like those gravy boats, which appeared in previous slides here. So those gravy boats allow the eggs to float at the surface while the water column is being poured out of the spout and replaced by clean seawater. So you're pretty much discarding all of the unused sperm in your culture. So this is, as I mentioned earlier, because we're rinsing quite early, this will allow you to handle embryos while they're not ongoing their first cell divisions, which you can see in figures B and C. D.lab, this is important to note. So D.lab is really prone to embryo breakage. That's what we call polyembryony, whereby embryos break into smaller but viable clonal embryos. We know that those smaller embryos do develop in normal larvae. They can also settle. It's, however, unclear that if, because of their smaller size, if their survivorship and growth after they settled might be jeopardized. So overall, we really try to avoid embryo breakage as much as possible in the culture by limiting handling during those stages from B to F. Okay, so as I mentioned earlier, embryogenesis in this species goes quite fast. So everything from first cell divisions to morula and gastrula will happen overnight, so on the first night of spawning. And basically the next day when you will come back to the lab, your embryos will have terminated gastrulation and will be rounded again. And during stages B to F, those embryos are extremely sensitive to breakage, gastrulation even more so, and we really recommend keeping them at low density and limit handling. However, during that period of time, we do recommend checking for fertilization rates. So fertilization rates will really give you a good understanding of how well your culture will be doing in the coming few hours. Because basically the eggs that were not fertilized in the coming hours will start, well, they're not viable, so they will start decaying, and they potentially can contaminate your culture. So the higher fertilization rates means that you have, first of all, more embryos to work with, but it also means that you'll have less decaying eggs in your culture and therefore less maintenance to do. So now if we go to the last part of this slide and look at the next day. So what do you expect on the next day following spawning? Again, those embryos will have finished gastrulation. They will be rounded again, which means that they are less prone to breakage and less sensitive. At that point, they should also still be buoyant. And that gives you, this is really like the best time window that you have to do one big complete water change. We actually, again, suggest doing a water change using fat separators, those gravy boats that I showed earlier, which allow the water column to be exchanged without discarding the eggs, or the embryos, sorry. So this is really important because if you manage to do that cleaning during the time window, you'll be discarding all of the unfertilized eggs that have decayed and do a really nice water change. But this time window is quite short. So a few hours later, those larvae will have reached motility and they will be swimming everywhere in the water column, which means that you no longer will be able to conveniently use those fat separators or gravy boats. And then you'll have to track every single larvae with the pipette to transfer it in a new water, or that you'll have to use a sieve, which, if it's done improperly, can be very damaging to those larvae or embryos. So this time window is really important. It will really give you a head start on your culture if you do it at this specific time. So this next slide here illustrates the timeline of larval settlement once those larvae have fully developed into swimming larvae. So again, this progresses very fast. Settlement competency will happen, again, really rapidly. So about 2 days after fertilization, larvae will be ready to descend. They will have already descended in the water column, and they'll either be lying or crawling on the bottom in search for settlement cues. On the third and fourth days after fertilization is really the time window where you would obtain the highest settlement rates if you provision your larvae with settlement cues at that time. So that's really when you want to be ready to add your settlement substrates. So basically, as you thought you were just about to take a breather from all of the culture maintenance you've been doing in the past days, you actually have to start setting up settlements for this species. So practically, take-home message from this slide, make sure you have conditioned substrates readily available for settlements. That will happen really quick. A small additional tip from Skylar Snowden, who in 2012 found out that adding a little bit of water flow, gentle water flow in your settlement containers will optimize settlement rates. And at last, be aware that most D.lab larvae will, they really like cryptic habitats, so underhangs, dark environments, and basically you will normally find them on the undersides of settlement surfaces. So again, here, no panic. If you don't see any settlers on your substrates at a first glance, they probably are all hiding underneath your tiles. Okay, so hopefully at that point you've raised hundreds to thousands of D.lab settlers, and you might be wondering, okay, what now? So how do we keep them happy and healthy and growing? Basically, processes that are driving early post-settlement growth and survivorship in corals are still quite understudied and constitute one of the CRC's focus topic, the CRC's larval propagation group. There are many environmental factors that can act independently or in synergy, together with also biological factors that influence the health of those coral settlers. And then on top of that, each species is also very different with different requirements, which makes this some sort of big jigsaw puzzle. But that said, we still know quite a lot of things. It's not all unknown. And here I'm going to give you the intel about what we know for D.lab. So first of all, what to expect in terms of growth or development after those corals have settled is that Diploria labyrinthiformis in the first months of its life, rather invests in polyp growth than polyp divisions. So that means that one primary polyp would just start growing in bigger size and will not divide all the way to almost 6 to 12 months. So in this picture here, this Diploria has only initiated its first polyp division after 10 months. This is quite contrasting with species that some of us might be more accustomed to working with. So for example, Acropora pomata, after a month, has already gone through multiple polyp divisions and is investing more in some sort of two-dimensional growth, so gaining space on the substrate rather than growing in larger individual polyps. So this means that you shouldn't worry if it takes forever for your Diploria settlers to start budding. That's just what they do. Secondly, we also know from a study that I conducted on Curacao in 2012 that the timing of symbiont acquisition in this species has a really big impact on their survivorship and growth. So here, the settlers that had acquired their symbionts within 3 weeks after settlement had an almost 4 times increase in survivorship in the following 5 months after they were outplanted on the reef. These settlers had also a 2 times higher growth rates than settlers that didn't have any symbionts at the time where they were outplanted, so at the age of 3 weeks. So it really seems like ensuring symbiosis early in the species by providing symbiodinium to Diploria labyrinthiformis is a potential very good way to optimize the culture of settlers. At last, we also have some information about the onset of heterotrophic feeding in this species. So these data were generated by Rob-Lucian Geertsma from the University of Wageningen on Curacao in 2018. And so Rob found out that D.lab primary polyps are able to capture and actually completely ingest Artemia nuclei as soon as 4 days after metamorphosis. And that by the age of 10 days, almost all of them, or 80%, 85% of those settlers were also capable of feeding. So this really suggests that feeding early in life for the species might be really important for them to sustain their metabolism and their health. So based on all of these different presentation and slides, you might wonder, what are the next steps? Well, first and foremost, we really hope to have motivated you to start working with the species, whether it's to help doing some monitoring work or to do some larval rearing and outplanting work with the species. As for more specific plans for Anya and I, so on Curacao this May, or next week, I should say, this is coming up fast. Together with project lead Kelly Latin-Howers, we'll be investigating the potential of optimizing rearing of those settlers by providing them early with symbionts and artemias in our in situ setting, so within SICOR's in situ larval rearing pools. We really hope that with these different protocols, we might be able to increase their growth and survivorship, and will at the same time optimize outplanting success. For Anya's team in Mexico, they're planning on investigating alternative sources of food, such as, for example, nutrients and amino acids. And her lab will also continue doing some cryobanking of sperm of this species to preserve the genetic diversity of Diploria labyrinthiformis, especially in the face of the stony coral tissue loss disease, which sadly in Mexico and many other regions of the Caribbean has dramatically affected this species. In conclusion, I would like to share with you a new concept that we've called the coral species fact sheets on the reproductive biology, early life history, and larval propagation of Caribbean coral species. So during this meeting or this webinar, Anya and I provided you with a lot of information. You might feel overwhelmed, or maybe you're afraid of forgetting stuff on the night of spawning. Well, don't worry, we've actually compiled all of this information in a really compact format in those fact sheets. And we will make the fact sheet for Diploria labyrinthiformis available on the CRC's website very shortly. For sure before spawning next week or the week after. There's, as mentioned by Anya, there's also a lot of other materials that will be available on our websites that will include at some point the recording of this presentation, as well as a PDF version of this presentation. All predictions, calendars, guidelines for monitoring, templates for data collection, and also not forgetting the 2017 webinar that will all be made available on our website. So I will now pass on the microphone back to Anya so we can start answering your questions. Thank you for attending these presentations.
Thank you, Val. That was really fantastic. We have some really interesting questions. A couple I have tried to answer when the information was available from my lab, or something very easy to answer. The first question that we have that came up is from Natalie Gutierrez, asking about, is D.lab split spawning a partial spawning? Partial being some parts of the colony, or does the whole colony spawn 3 times, i.e. 3 bundles release by polyp in the year? Val, would you like to answer that question?
Yes, sure. So split spawning is typically when you have like a population of corals that only one part of the population will spawn on a specific month, and then the next month you'll have the other part of the population spawning. This can also be the case if individual colonies are partially spawning on one month and again partially spawning on the other month. And that usually happens when the full moon might be a bit confusing. So there are some months in the year where the full moon might happen both at the beginning and at the end of a month, which means that corals might not all agree, so to speak, on the month where they should all spawn. So basically all the reproductive efforts becomes a bit more spread throughout the year. For the case of Diploria labyrinthiformis, what we see in those pulses is really like on one specific evening during, let's say, like a time window of half an hour, there will be multiple pulses of one area of that colony. Quite often, we'll see that maybe if that same colony spawns again the same day, it might spawn from another area on that same colony. So, but that is not necessarily, it's not really split spawn. That's just like one colony that's spawning in different areas over different nights.
Yeah, we get a similar situation in Puerto Morelos. Some tagged colonies will spawn on different months, successive months, and it'll be either different parts of the colony generally, or different colonies within the same population. So thank you for that question, Natalie. The next question came from Ana Carolina Hernandez, who asks, what would be the correct way to cite the Caribbean-wide D.lab spawning monitoring information? I have answered her, but I think it's something that we should talk about here in case anybody else would like to cite it. My suggestion is to contact the person responsible for the data collection and to ask directly, because most of the data that we have presented today may not yet have been published. Would you like to add anything else, Val, to that?
No, I think that's a very complete answer, Anya. Thank you.
Okay, the next question comes from Frida Barron, who asks, how does full moon influence spawning? Would you like me to answer that, Val, or would you like to?
Sure, go ahead.
Okay, so basically there's been some very interesting papers that have come out about this. Oren Levy in 2007 published a paper showing that some corals at least have cryptochromes, and so these might be sort of receptors for moonlight. And so when there's a full moon, the coral will basically detect the brightness of the moon, and then a whole series of molecular processes will then engage and result in synchronous spawning. There's also been some work by Gorbunov and Palkovsky in 2002, basically showing that corals do have a blue light sensor, even though they don't have per se light recepting organs. So, but we really don't understand the molecular detection of moonlight yet, but basically it's most likely that cryptochromes are involved. Anything you'd like to add, Val, to that?
No, that was fantastic. Thank you.
Okay, the next question from Hanais Batias. I hope I pronounced your name correctly. If not, please forgive me. She's asking about Pseudodiploria strigosa and clivosa, which have been reported to spawn in fall and autumn. And have we seen these two species spawn in the spring in other locations? I can just let you know very quickly in Protomorelos, we've only seen them spawn in July and August, and not always in August. So I'm sure, Val, you have more information about those two species.
So personally, the data that we have, at least for Curacao, is pertaining only to strigosa. I have personally never monitored clivosa. But what I know for strigosa is that we've seen them the earliest in, that was in early July, all the way till October. There is actually quite an interesting thing in this species is that we've seen sometimes really early spawning in terms of times after sunset. So something around like 20 to 40 minutes after sunset in some corals or some colonies, and then other colonies actually spawn much later in the evening, so somewhere around 10:30 or 11 p.m. This is something that we actually have included in all prediction calendars. There is information on that species actually available on the CRC's website. But there could be potentially another situation where there's two cryptic species in Diploria strigosa based on those two different timings of spawning during the evening. So yeah, that's as much as I can add for other brain corals or other Diplorias. Okay,
I'm just going to jump down here through a couple of questions because there's one person saying she can't see the questions. On the control panel on the right-hand side of the webinar, there should be a tag that says questions. And if you open that tag, you will then be able to see, to write your question in there. I hope that helps. Okay, so the next question is there a minimum size of D.lab colony needs to be to spawn, as well as how healthy does a colony need to be to spawn, i.e. percentage mortality or presence of disease? I don't have a lot of healthy colonies to survey. This is from Kimani Kitson-Walters. I'll just speak right now immediately for Puerto Morelos. We've had some colonies last year that had only 10% live tissue, and they spawned amazingly and had really good fertilization rates. Whereas we had another colony that had about 90% live tissue, and it spawned a little bit, not so much, and didn't have very good sperm quality. So I think it's really variable. But I think if you're dealing with diseased colonies, as Val said earlier in her talk, you really need to be careful about handling the nets so that you don't pass the disease from one colony to the next. Doing dips in chlorine, etc., would be a very good idea. And I guess just monitor the colonies. We try to work with healthy colonies preferably, but with SCTLD running rampant through our area, we were pretty limited this year as well. So good luck with that. Oh, and the minimum size from Ernesto Wales, I'm trying to remember the minimum area that a colony needs to be, and I can't come up with it right now.
It was 100 square centimeters.
Okay, which is not very big, really.
No.
10 centimeters.
We've seen quite small colonies spawn very successfully on Curacao, at least.
Yeah. Do you have anything to add about the disease?
No, I think you covered it pretty well.
Okay. Okay, question from Aldo Crocker. Is there any morphometric study and aggression study to support genetic population evidence? And are reproductive events independent, or is it a case of split spawning?
Hi, Aldo. I can answer your question. So I'll first start with the independent reproductive events and versus split spawning. So basically, they are independent reproductive events. So in the same year, you'll have colonies both spawn in the spring, and then a completely different set of colonies spawn in the fall. And as I mentioned, this is really consistent, and it's separated by 1 to 2 months of no spawning of those colonies between the two events. So those are really independent reproductive events. In terms of morphometric studies, we haven't been doing very detailed morphometrics. We were actually pretty nervous of sacrificing parts of those colonies to do those morphometrics, and really thought, okay, like, is this worth, you know, investigating further, or should we just rely on the genetics and the field observations that we have? So basically, that's still an open question. If potentially they have different morphometrics based on the different timing of reproduction, but we haven't pursued that question so far. Thanks for your question, Aldo.
Okay, next question from Avavan Devendiran. I really hope I pronounced your name correctly. I'm sure I didn't. But a good question is, do the larvae settle in any substrate, and are the larvae attracted to any specific colored substrate? Diploria for us settles on everything. That's not about for you.
I can
and
elaborate. So Diploria labyrinthiformis actually is quite easy to induce in terms of settlement, but there are still quite marked preferences. Normally we work with a CCA or a crustose coralline algae that's named hydrolithon, and that species of CCA has been shown by, for example, by Raphael Ritson-Williams' lab, that it induces settlement in many different Caribbean species. So typically, we really try to have our substrate conditioned in the presence of that CCA species, which really helps triggering much higher settlement rates than if substrates are conditioned in just like, you know, random setting. So yeah, D.lab is pretty easy to induce in terms of settlement, but there's really ways to optimize that. And that would be my answer would be to really use a hydrolithon species.
Mm-hmm. Okay, given from Rachel Seraphin, given their rapid bundle breakdown, did you see any percentage of self-fertilization as it related to time?
I can also answer this, and because it's something that we, like sort of on the side, we actually studied while doing experiments together with Tanya and Matthew, when we were looking into different fertilization success. So if you remember, when we looked into different sperm concentrations, so let me rewind a little bit. So even if bundles have broken up really early, and that sperm from the same colony might be in contact with loose eggs from the same colony for quite a long amount of time during the dive, after we got back to the lab and separated those sperm and eggs from the different parents, when we started doing different sperm concentration assays, we never noticed fertilization rates in groups where no sperm was added again. So this really leads to think that there's no, or at least not really high rates of self-fertilization in Diploria labyrinthiformis. Did that make sense? This was maybe a little weirdly explained.
Yeah, we also, just to back you up, Val, using our cryopreservation assays, we also don't find self-fertilization in D.lab. Okay, so the next question from Samia Sarkis. What is density of fertilized eggs for development to embryo? What is tank volume used? Well, it really depends on your facilities, but fertilized eggs for development to embryo. We actually go by surface area rather than by volume. How about you, Val? We do about, try to do about four eggs per milliliter. Well, I guess per centimeter squared, which will be per milliliter.
Yeah, so in general, we really try to keep our culture as as lowest density as is possible in terms of space that you have at your facilities. One general rule of thumb is that you really don't want to have more than one egg or embryo per milliliter. And that might sound very small, but it really will give you a big heads up on your culture if you keep them at low density. So basically, when you are after you rinse your eggs and you're transferring them in multiple other containers with filtered seawater, the rule is if you think it's a low density, you probably want to go even lower. And I've been really really learning that way, and it's proven to be really useful to keep them at low density. And that's really something I have to mention here. But Mark Vermeer has been like really pushing for this, and it's been every year he comes and inspects the cultures and makes sure that everything is at low density, and it really, really works. It saves you a lot of time later on, and it also saves a lot of embryos.
Yes, it's true. They get anoxic very quickly, especially if you get bacterial buildup. So, but for example, the tanks we use are about, they can hold 80 liters each, and we have about, but we only fill them up about 50 to 60 liters, but we tend to go by surface area rather than by volume, especially because the fertilized eggs are floating at the time.
Yeah, exactly.
So what substrate is the best for the larvae to settle? Depends on the species for us, but for D.lab, they do like cement sand mix. They do like ceramics. Val, what have you found?
I think that one thing to keep in mind with this species is that they really like settling in more cryptic habitats. So cryptic meaning, low light environments where they're more sheltered from later on, when you put them on the reef, that they're more sheltered from competition and predation. So you want to have substrates that in some way allow larvae to crawl underneath and settle underneath, but also that when you're going to be transferring them to the reef, if that's your goal, that you're not going to be crushing them, by fixing those substrates on the reef. So making sure that there's some sort of hidden or low light habitat on those substrates is really important for the species.
As it is for a number of species.
Yeah, for many species actually.
Louis Pierre Rich says, I have some D.lab histo data that could possibly be useful. Who should I reach out to?
Well, that's amazing.
That would be really great.
You can email them. My email address is on multiple slides of this webinar. Otherwise, it's v.chamberland@ccor.org. But yeah, that would be really amazing if you could share that information with us. We would like to know more.
Yeah, awesome. Thank you very much, Louis. Okay, from Cheryl Hankins, are there any data or do you know of D.lab spawning in a laboratory setting? I know some people want to try this summer, especially because of the SCTLD disease, but I don't know if at Moat they have any D.lab. Do you? Val, do you know if at Moat they have any?
I think they have D.labs, but I don't know of any XC2 observations of spawning. I do know that there's, from the AZA network, and I think there's potentially a lot of people from that network on the line today, that there are people that are in the rescue program, but because of the stony coral tissue loss disease, cannot say this, but that have Diplorias in their tanks, and maybe this year will be the first time that there's going to be some sort of common effort of monitoring the species XC2. So we don't know much at this point, but hopefully this year will get us more information on the species XC2.
Good. Okay, so from Nicole Fogarty. Hi, Nicole. She said, does D.lab self? So we think it doesn't.
Yeah, Nikki, I think that from many trials that we've done, and including a lot of controls with no sperm addition and so on, we haven't seen any really large evidence of selfing. So compared to like a lot of other species can do selfing, for D.lab it doesn't seem to be a very common thing.
Okay, next question from Sobeida Nunez. She says, what kind of bag should we use for collecting eggs during spawning? Well, we use, it's not really a bag, it's a tent that's made from very cheap kind of netting. I can send you the way that we make our nets. And I think, Val, you also have used sort of tarpaulin type nets. Is that for D.lab?
Yeah, so we've been trying different types of nets. And actually, I think the best way to answer this question is to direct you to the 2017 webinar, where we do have complete instructions for building different types of nets. And that webinar also includes like the pros and cons, or like situations where you want to use which net for what reason. So that will definitely depend on conditions at your site, size of the corals, etc. So yeah, I think the best material to answer this will be the 2017 webinar, which is again available on the CRC's website, as well as the Reef Resilience Network's website.
Yeah, excellent. And I can also send you the manual that we have that shows how to build nets if you need.
Great.
It may be possible in the future to make symbiosis associations in the laboratory. Yes, yes, I think a lot of labs have been able to initiate symbiosis of organisms that that are symbiotic at some point in their life cycle, and then can reinitiate symbiosis. I think that can be done in the laboratory. I mean, we can have the recruits that you know, do not have symbionts associated with them, and give them symbionts. They can be cultured, or they can be extracted from a host tissue and given to the recruits, and they'll take them up. Would you like to add anything else, Val?
Sorry, I was looking for my unmute button. No, I think you summarized it pretty well. I just saw in the question box that Valeria Pizarro from TNC in the Bahamas, she actually just added information about XC2 spawning at Cape Lutheran Institute, where they actually did have XC2 spawning, but only from one of their colonies. And they also observed self-fertilization, but those actually yielded few settlers, which at the end didn't survive. So it might be that self-fertilization might occur in some circumstances when there is no other options, but that those embryos or larvae are not the healthiest ones. Mm-hmm. Thanks, Valeria, for adding information.
Okay, so Ayumi says the monitoring for the Windward Islands, being Saba, St. Eustatius, St. Martin, and a few others, do we follow the moon schedule provided under Dominican Republic? Well, what we tried to do during the seminar was to give a really broad spawning window that I think should cover that area. So yes, I think you should follow not necessarily Dominican Republic, but the next slide down where I sort of gave the summary, which is 9 to 14 days after full moon. I would start there and then try to narrow it down with more observations in your particular area. Anything to add there, Val?
Yes, I wanted to add, so this information session, well, basically, Anya and I were a bit surprised that there were going to be so many people on the line. We first thought this was going to be a very informal session where we could go over like very detailed, you know, how to calculate spawning times and how to make your prediction calendars, how to use the templates. We haven't done that today because we probably would still be sitting here for another hour. But for those who are really interested in doing monitoring, we would be very happy to organize another smaller side session where we'll go over all of these specific questions together. So please, if that's interesting to you, email us. We will also be sending a poll after this webinar, not today, but somewhere in the coming days, to all of those who have registered. And then we'll be asking that those who are interested in doing monitoring reply back to us so we can form a smaller, so we can add you to our monitoring network across the Caribbean.
Okay, I think we've reached the end of our webinar, which is till 3:30. So are we able to go on, or do we answer the rest of the questions by putting them up and putting them on the website?
I think, Anya, for the remaining questions, because there's still a lot of what we can do is, we will answer them after this webinar and we will post them on the CRC's websites. So look for them shortly. We probably will be able to also send you guys an email notifying you that we have answered your questions and giving you the link to those answers. Wow, this was amazing. Thank you so much for everyone's interest.
Yes, we really appreciate you taking the time to listen to us and to these really great questions that you've asked us. And can we go to the next slide and just say thank you and stay safe and healthy?
Yes, there it is. I thought you were still in control, so my bad. But yeah, thank you so much.
It hasn't yet changed over here.
Okay, well, this is what concludes this webinar. Again, thank you everyone for joining in so many numbers. And yeah, please stay safe and healthy, take care of yourselves, and hopefully talk soon about more monitoring and D.lab work.
Yes, and we're available to answer any questions by email. You have both of our emails in the webinar. Thank you. Stay safe.
We are, last tiny notes. We're also interested in spawning observations for other species. So if you want to be part of monitoring efforts for other species in D.lab, also please reach out. We are, as Anya mentioned, we're building a database for spawning observations of all Caribbean species, but also Indo-Pacific species. So if you're out of the Caribbean region and want to contribute to this type of data sets, please get in touch with us. Thank you.